हेलो, हाय गाइस। सो आज का हमारा टॉपिक होगा कल्चर मीडिया। माइक्रोबायोलॉजी में स्टार्ट करने के बाद जनरल माइक्रोबायोलॉजी का एक बहुत इम्पोर्टेंट पार्ट है कल्चर मीडिया, क्योंकि ऑब्वियसली कल्चर मीडिया इज़ वेयर हम लोग बैक्टीरियाज को ग्रो करने वाले हैं। और लेट्स स्टार्ट विद द डेफिनेशन ऑफ कल्चर मीडिया। व्हाट एक्चुअली इज अ कल्चर मीडिया? सी, at the very basic, culture media can be liquid as well as solid. That's not right. Can be liquid or solid. ये misconception बिल्कुल भी नहीं रखना कि क्योंकि ये culture media है तो it's always gonna be solid. It can be liquid as well. So let us define a culture media. Culture media is a liquid or solid substance which contain nutrients to support the growth and survival of microorganisms. So these are a liquid or solid substance to support the growth. Support growth. किसका growth? Of microorganism. So microorganism की growth को support करने के लिए जो liquid या solid media हम लोग use कर रहे हैं, which contain nutrients as well, is known as microorganism. Culture media. Growth and survival. Support. And ये support कैसे मिल रहे हैं? Because they contain nutrients. They have nutrient and they support the growth and survival of microorganism. अब constituents of culture media पे आते हैं. कल्चर मीडिया का कॉन्स्टिट्यूएंट्स में जो बहुत बेसिक चीज़ जो रहने वाला है इज फर्स्ट थिंग इज गोइंग टू बी वाटर. सेकेंड is electrolyte. The third will be, let's say, peptone first. Peptone is actually a complex mixture of partially digested protein. Partially digested protein का complex mixture is peptone. And then there is agar. It is actually a solidifying medium. मतलब हमारे कल्चर को जो थोड़ा सा सॉलिड टाइप का कंसिस्टेंसी देगा उसके लिए वी आर यूजिंग अगर हियर. सो जस्ट द मेन थिंग टू कीप इन माइंड अबाउट अगर इज दैट इट इज अ नॉन न्यूट्रिटिव, मीन्स इट हैज़ नो न्यूट्रिटिव वैल्यू. इसका कोई न्यूट्रिशन में रोल नहीं है. इट इज जस्ट टू कीप योर मीडिया इंटैक्ट, गिव इट अ कंसिस्टेंसी, मेक इट सॉलिड. देन अपार्ट फ्रॉम दिस हम लोग न्यूट्रिशन के लिए क्या यूज़ कर रहे हैं? तो वो मीट एक्सट्रैक्ट हो सकता है, it can be yeast extract, or it can be blood. If you are using blood, definitely you can use serum also. So these are the 7 constituents of culture media, including water, electrolyte, peptone, agar, meat extract, yeast extract, and blood or serum. Now, what are the types of culture media? Culture media को हम लोग 2 वेज में क्लासीफाई कर सकते हैं. Let's write the classification. So there are 2 ways. First, I'm gonna say, based on consistency. Based on consistency, ये तो आप लोग समझ ही गए. It can be solid, it can be semi-solid, और it can be liquid. Liquid यहाँ पे broth भी हो सकता है, which is not semi-solid but not liquid also. So we did broth. And then the second classification, it can be based on the method of growth detection. It's not that important, so we're not gonna go in detail of that method of growth detection. I'm just gonna tell you that के बेसिस पे हम लोग इसको कन्वेंशनल में डिवाइड कर सकते हैं एंड ऑटोमेटेड में डिवाइड कर सकते हैं। नाम से ही पता चल रहा होगा, कन्वेंशनल आर द वन विच इज़ प्रिपेयर्ड फ्रॉम न्यूट्रिशंस, जैसे कि एक्वस, एक्सट्रैक्ट्स ऑफ मीट, पेप्टोन, ये सब यूज़ करके वी आर मेकिंग द conventional one. So the bacterial growth is detected manually. We are doing it manually by visual inspection of turbidity and colony morphology. And the automated, it is like uh, the growth is detected automatically by equipments. इसमें हम लोग machines and the equipments use कर रहे हैं. We are not doing it manually. So ये दो टाइप का हो चुके हैं classification. We are done with classification. Now let's get started with conventional culture media. There are many and many conventional culture media. क्योंकि हम लोग अभी स्टार्ट कर रहे हैं, so we're gonna start with the basic one, which is the simple or basal media. सिंपल और बेसल मीडिया से ही पता चल जाओगे कि ये सबका बेसिक है। ये मिनिमम इंग्रेडिएंट कंटेन करता है और ये सपोर्ट करता है ग्रोथ ऑफ नॉन फास्टिडियस बैक्टीरिया। इट हैज़ द मिनिमम ingredient plus it support non-fastidious bacteria. Now, what it actually contains? बोल तो दिया हमने कि it has minimum ingredients, but what does this minimum ingredients include? So first I'm gonna say is peptone water. वाटर एंड इलेक्ट्रोलाइट वो बेसिक है, वो हम लोग बार बार नहीं लिखेंगे। उसके अलावा क्या होगा, दैट गोना बी दिस व्हाट वी गोना टॉक अबाउट। सो पेप्टोन वाटर का वैसे यू डोंट नीड टू रिमेंबर इट, बट जस्ट फॉर योर नॉलेज, पेप्टोन वाटर में देयर विल बी 1% पेप्टोन. Do you remember what is peptone? Peptone is a partially digested protein, like I said earlier, and it's gonna contain NaCl 0.5% plus water, obviously. So this is what peptone water contains. You don't have to remember it, but now let's get forward to the second constituent. Second constituent is nutrient growth. क्योंकि हमें न्यूट्रिएंट प्रोवाइड करना है, सो ऑब्वियसली वी नीड समथिंग. न्यूट्रिएंट ग्रोथ, व्हाट इज एक्चुअली न्यूट्रिएंट ग्रोथ? न्यूट्रिएंट ग्रोथ, अगर हम लोग पेप्टोन वाटर में मीट एक्सट्रैक्ट मिला दे, हाउ मच? 1%. पेप्टोन वाटर में 1% मीट एक्सट्रैक्ट मिलाने के बाद यू गोना गेट अ वेरी फ्रेशली मेड न्यूट्रिएंट ग्रोथ. नाउ ये भी 3 forms क्या होता है? It can be of 3 form. What are these 3 forms? It can be extract, but let's write meat extract, okay? It can be an extract. It can be an infusion, and it can be meat digest growth. You don't have to remember all those. Now coming to the third one, which is nutrient agar. जैसे कि आपने पहले देखा होगा, I didn't write agar before this. It was broth, so definitely it's going to be broth, liquidy kind of, not solid or semi-solid in any form. So now that we are here in agar, तो obvious है nutrient growth में अगर मिलाना होगा, how much? It's 2%. 2% is to make it solid. And if you want to make semi-solid media, then you can reduce it from 2.5 to 0.5 to 0.2%. That's gonna make it semi-solid. Now, basal media का constituents हमने पढ़ लिए. What's the basic thing about it is that it's a basic. It has minimum ingredients and it allows the growth of non-fastidious bacteria. पेप्टोन वाटर इसका पहला कॉन्स्टिट्यूएंट है. पेप्टोन वाटर हैज़ पेप्टोन, इट हैज़ NaCl and it has water. Now meat growth or nutrient growth. Nutrient growth is peptone water plus meat extract, which means it's gonna have peptone, NaCl, water plus meat extract 1%. 3 फॉर्म्स में हो सकता है, मीट एक्सट्रैक्ट, मिक्स मीट इन्फ्यूजन एंड मीट डाइजेस्ट ग्रोथ. थर्ड इज द न्यूट्रेंट अगर को इट्स लाइक न्यूट्रेंट ग्रोथ में अगर मिला दो 2%. सो ये सेम केस में हम ऐड करते जा रहे हैं, वन मोर ingredients we are adding to the previous one and getting something new. Now what is the use of basal media? Sorry, I got this. Now the use of basal media. Basal media, these are used to test the growth of non-fastidious bacteria. as हमने पहले ही देखा कि basal media allow करता है growth of non-fastidious bacteria. They serve as the base for preparation of any other media. कोई भी नया मीडिया बनाना है तुमको तो इसको हम लोग एज अ बेस यूज कर सकते हैं. मतलब यू कैन कीप ऑन एडिंग न्यूट्रिएंट्स इन दिस वन एंड गेट अ न्यू मीडिया, राइट? Now it can be used for the study of bacteriological growth curve. Bacterial growth curve, I'm hoping that most of you would have studied about it. The growth curve वो कुछ ऐसा दिखता है that it has log phase, then it has stationary phase, and then it has not it has log phase, then it has stationary phase, and then it goes into death, kind of all those things. Okay, let's not go there. So now after this, the nutrient agar is the preferred media for performing the biochemical tests like oxidase, catalase, side agglutination test. ये सब को परफॉर्म करने के लिए we choose nutrient agar. Okay, nutrient growth को हम लोग bacteriological growth curve के लिए यूज करते हैं. Semi solid media they are used for motility test. Semi solid is used for demonstration of motility test. Now the Selective Media or the Basal Media is finished, so we're gonna step into the second kind of media, which is Enriched Media. Are you guys getting my handwriting? Please tell me if it's like not that clear. So Enriched Media. अब क्योंकि हम लोग प्रीवियस स्टेप में एक स्टेप आगे कुछ इंग्रेडिएंट्स ऐड करके आगे बढ़ लेते हैं, सो वी गोना डू द सेम. Enriched media is nothing but when you add basal media with some additional nutrients. ये न्यूट्रिएंट क्या हो सकते हैं? अगर हम लोग पहले जाके देखें द कॉन्स्टिट्यूएंट्स, तो हम लोग मीट एक्सट्रैक्ट ईस्ट एक्सट्रैक्ट हम लोग पहुँच गए थे, बट डिड वी रीच द ब्लड एंड सीरम? नो. So what is the purpose of blood and serum? We're going to use this blood and serum as a nutrient in this enriched media. Yes, the nutrient in this enriched media is going to be blood or serum. Or it can be egg also. Egg yolk, it's used. It allows the growth of non-fastidious. They also support the growth of fastidious nutritionally extracting bacteria. So जैसा हमने previously देखा, basal media was for non-fastidious bacteria, but it is also for fastidious bacteria. The example. Let's get into the example of enriched media. The first example is blood agar. I'm not gonna write everything, but I'm gonna tell you about blood agar. What does actually blood agar contain? So it's prepared by adding 5 to 10% of sip blood to the molten nutrient agar. Molten nutrient agar, you know nutrient agar. Here are nutrient agar which is nutrient growth plus 2% agar. उसमें अगर हम लोग शिप का ब्लड ऐड करते, take nutrient media, melt it at 45 degrees Celsius, and then add 5 to 10% of sip blood and your blood agar is ready. It is most widely used media for the diagnostic test of in bacteriology. So blood agar test में हम लोग क्या कर सकते हैं? It has a hemolytic property. If you know, like if there is blood, so obviously there is going to be hemoglobin, right? And it has a hemolytic property. Some of the bacteria, they can hemolyze the blood. So उसके basis पे we can differentiate, like the bacteria which can differentiate the blood agar partially, completely, like that. So the test, the hemolytic property of the bacteria allow us to do this test. इसके basis पे it can be of two type, partial or alpha hemolysis. Alpha hemolysis or part is actually partial hemolysis. Here in this condition you'll get green color in around the hemolysis, whereas complete or beta hemolysis, we'll discuss it further. Now the second kind is chocolate agar. Chocolate, yeah, na? Seems tasty, right? But it's not. You're not supposed to eat that. But we are supposed to read about it. Let's read about it. Chocolate agar. Chocolate agar is heated blood agar. फिर से previous step में कुछ ना कुछ ingredients है उसको modification करके we wanna move forward. So chocolate agar is a molten or heated blood agar which is prepared by adding 5 to 10% of sick blood to the molten nutrient agar at 70 degrees Celsius. Mind you, last time we were heating at 45 degrees Celsius. This time we are heating it at 70 degrees Celsius. So the RBC, it will sterilize and the content of RBC will be released, जिसके कारण यहाँ पे chocolate agar में RBC is already lysed, okay, by heating it at 70 degrees Celsius we have lysed the RBCs, जिसके कारण the contents of the RBC is released and the color has changed now from red to brown. And so does it look brown. Now it is more nutritious than blood agar and even support the growth of highly fastidious bacteria. It support the growth of highly fastidious bacteria. Now अब highly fastidious bacteria के बारे में जिक्र कर चुके हैं, तो let's give one example. One of the highly fastidious bacteria is H. influenzae. Yeah, it does not grow on blood agar, but it does grow on chocolate agar. Now the third example of enriched media is Loeffler serum slope. Loeffler serum slope इसलिए बोलते हैं क्योंकि when you keep it in this test tube, it makes a slope kind of. So yeah, it's Loeffler serum slope. What's Loeffler serum slope? Yes, क्योंकि इसके नाम में सीरम है, it contains serum. Previously we were using blood. Now here we are using serum. It is used for isolation of, please remember this, that it is used for isolation of Clostridium. Really sorry. It is used for isolation of Corynebacterium diphtheria. Corynebacterium diphtheria. Spelling में थोड़ा सा mistake है, sorry. It is Corynebacterium diphtheria. Right. Please remember this. You can be asked this in your MCQs, in your Vivas. Now the fourth one we are coming up with is blood culture media. It is also called Enriched Media for isolation of microorganism from blood. And let's see. Now we are done with Enriched Media. Let's revise it again. Enriched Media is actually basal media plus the nutrients. Nutrient can be blood, serum, or egg. It allows the growth of fastidious bacteria. The examples are blood agar. Blood agar contains the ship blood, 5 to 10% ship blood, which is heated at 45 degrees Celsius. Now it give three type of hemolytic test. Sorry, not three, two type. It's alpha hemolysis and beta hemolysis. Alpha hemolysis is a partial hemolysis. Whereas beta hemolysis is complete hemolysis. Coming to chocolate agar. Chocolate agar is highly fastidious bacteria. It allows the growth of highly fastidious bacteria. ब्लड अगर को हम लोग हीट कर देते थे at 70 degrees Celsius, which allows the complete lysis of RBC, leading to the change in color of blood agar from red to chocolate brown. So here we name it chocolate agar. It also allows the growth of highly fastidious bacteria. One of them is H. influenza, which doesn't grow in blood agar. Third one is Loeffler serum slope. It is very important to remember the example, which it is used for the isolation of bacteria, Corynebacterium diphtheria. And the fourth one is blood culture media. It is for isolating microorganisms from blood. Coming to the third type of culture media. Let's change the color of our pen. Let's go with blue. Do you want blue? Oh, white. Let's go white. So the third is enrichment probe. No, don't confuse it with enriched media. They're not the same. They're quite different. Enrichment growth is actually a liquid media which is made with some inhibitory agents and selectively allow the growth of certain organisms. It is liquid media in which we add some inhibitory agents. Now you're gonna ask me, with culture media, the main purpose of culture media is to grow the organism, right? So why are we adding inhibitory agents here? Taki we can differentiate between different bacteria. So if like there is a type A bacteria which cannot tolerate bile, suppose, and you add bile in this media, so do you think A will grow here? No. If the A doesn't grow in this culture media, then it's gonna be kind of indication that this is the culture of bacteria A. This was a short example how we work around it. So this inhibitory agent, what it does is that it selectively allows certain organism to grow and inhibit the others. Now let's give you some example. First example is gram-negative growth. Ever heard of it? Yeah. Same around gram-negative bacteria, gram-negative growth. It is used for. Let us remember one example also. It is used for the isolation of Shigella. Second one we're gonna come around is tetrathionate growth. The tetrathionate growth is used for Salmonella typhi. S. typhi causing enteric fever, very familiar, right? Then there is selenite growth, which is again used for Shigella. So you're gonna remember for Shigella we use bacterial gram-negative growth and selenite growth. For S. typhi we use tetrathionate growth, and there is also an alkaline peptone water, which is used for Vibrio cholerae. You're gonna need this in your further chapter where you're gonna be reading about cholera and the diarrheal diseases. All these medias are gonna come up there once you do the lab diagnosis for this particular organism. Then you'll see that why we are reading about this culture media and why do we need to grow this bacteria. Because it's gonna tell you and it's gonna help you diagnose the disease. Right? Now let's come to our fourth culture media, which is selective media. नाम से ही समझ में आ रहा होगा. It contains inhibitory substance which inhibit the normal flora present in the specimen and allow the pathogen to grow. So what it doesn't allow is normal flora. जो हमारे गट में जो normal flora है उसको ये नहीं allow करेगा, and what it allows are the pathogen. So only the pathogens are gonna grow in this media. Let's see the example. a very famous Lowenstein Jensen media, also known as LJ media. It is used for the isolation of Mycobacterium tuberculosis. Please remember the examples. Winston Jensen LJ media is a selective media which is used for tuberculosis. इससे पहले we talked about Loeffler serum slope, which was used for Corynebacterium diphtheria. Do not confuse in between them. Now, the second example is TCPS. It is thiosulfate citrate bile salt sucrose. Let me write it for you. Thiosulfate citrate bile salt sucrose agar. It is used for Vibrio. What was the other media which we studied in the previous one? The enrichment broth, which were used for Vibrio. It was alkaline peptone water. Now, the third example is DCA, which is deoxycholate citrate agar. Deoxycholate citrate agar and XLD as well. XLD is xylene, lysine agar. It is used for Salmonella and Shigella found in the stool. So you're getting it right. What it's gonna do is it will allow this DCA or XLD xylene lysine agar is gonna allow the growth of Salmonella and Shigella and not the normal commensal bacteria or the gut flora. Like you can say E. coli or Lycobacterium or these things are not gonna grow. The commensal bacteria, they are not gonna grow in the medium. Also, there is one potassium telluride agar which is used for Corynebacterium diphtheria. Now let's come to the fifth one. The fifth, what's in the color again? This is the transport media. Now the color is clear. Do you want the pen to make it like more thick a bit? Is it fine, right? So yeah, transport media ना इसके भी नाम से पता चल रहा होगा. We use it to transport the clinical specimen. एक जगह से दूसरे जगह ट्रांसपोर्ट करने के लिए what we use is this transport media. But what's different? How is it like different from the other bacteria? So the main role or the main where we need this transport media is the characteristic feature of this transport media. It is to contain the delicate organism when there is delay in transport. Here in this media, the bacteria does not multiply. Remember this very important point. The bacteria does not multiply, but it remains viable. Means you're gonna let them stay viable, but you won't let them to grow. If there is one bacteria in the culture, whatever you were carrying, when you reach to the hospital from that point, there is going to be one bacteria and is going to be alive. Bacteria cannot grow into two bacteria, no. We can't allow that because it will falsify our result, right? Now the example of this transport media we can see is autoclave seawater. Then you can see Caddy Blair media. You can also use these two are used for Vibrio cholerae. Others are buffered glycerol saline. It can be Enriched Media, Shorts Media. There are many examples. Let's just remember two for now. Now the sixth one is Differential Media. Do you remember we read Differential something ahead? What was it? So this differential media is going to differentiate between the two groups of bacteria by using an indicator. So what we use here is an indicator to differentiate between two bacteria. And it changes the color of the colony of the particular group of bacteria, but not the other. Like you have got group A colony, and then you have group B colony. Maybe if you grow group A in this culture media, it will give you red color. But the original color of this media was blue. So now if A gave you red color and B didn't give you red color, which means it didn't change the color of the media, so you can differentiate the media. It means you can, yeah, both these colonies are different, they have different property, one does differentiate, one doesn't. And you can recognize which of them does change the color, and you can like group them according to the color changing property, right? So what the first example here is McConkey agar. It is actually a differential and low selective media. Low selective, which means it is not that highly selective. Used for isolation of enteric gram-negative bacteria, it differentiates between lactose fermenting and non-lactose fermenting bacteria. So the lactose fermenting bacteria, or they are the lactose fermenter, they give pink color, cute pink color. You're gonna read about this lactose fermenter, but for now you can remember E. coli is a lactose fermenter. It gives pink color. Klebsiella is a pink color. Colony forming or lactose fermenter. Now there are some non-lactose fermenter like Shigella. What color does it give? No color. Its life is colorless, like us medical students. So yeah. So try about it in the next example, which is clade. Clade is cysteine lactose electrolyte deficient agar. Cysteine lactose electrolyte deficient agar. It is another kind of McConkey, and like McConkey, it can differentiate between non-lactose fermenting and non-lactose fermenting. Now the seventh kind of media we're gonna talk about is anaerobic culture. My throat is actually so dry. I'm so thirsty. Talking so long. This culture media, it reduces the substance which takes up oxygen and create a low redox potential. It takes up oxygen. Sorry, it's an anaerobic, right? So it takes up oxygen from the substance. Like whatever the substance you're gonna use, it takes up the oxygen and creates an anaerobic environment, and then it reduces the redox potential, and it allows the growth of obligate anaerobe. Example is Clostridium, which is a very classical example of anaerobic bacteria. The example include anaerobic culture bacteria is Robertson cooked meat broth. Sorry for this handwriting. I hope you will get me. Robertson cooked meat broth, it contains. The constituent is important here. It contains chopped meat particle. The meat particle here is beef heart. What does this beef heart do? So this beef's heart, it provides glutathione and unsaturated fatty acid. It is mostly used as anaerobic culture media, right? And the maintenance of stock culture is also used for that. Other examples are brain heart infusion, which is one of the most important culture media. Just like Robertson cooked meat broth. Now coming to the 8 or the blood culture media, just we just gonna name it, not talk much about it. It is the recovery of bacteria from blood is difficult as they usually present in lesser quantity in the blood. Hence, therefore enriched media are used for isolating microorganisms from blood. Now, with this, the conventional, sorry, the blood culture media is again divided into conventional blood culture, which include monophasic medium like inert infusion, or biphasic medium like it contains liquid phase containing BHI broth and a solid agar made of BHI broth. Okay. And the second is automated. Automated you don't need to know much about, but just remember the name. There is continuous automated monitoring. There are automated systems. These systems commercially which are available or mostly what you're gonna hear about is BACT, Alert 3D and BACTEC. There are also some disadvantage like they're highly cost equipments and they cannot observe the morphology of the colony as the liquid medium is used. And also they are beneficial as they are fast and not manual, less chances of any what do you say breach in the medium. So yeah, that's much too much about it. And sorry about today. That's all about today. Now we will pick up another topic, which most probably is going to be culture method, I guess, for the next class, next day. For now, thank you. Please revise your notes, read it again, and remember the important examples. Ta-da!